Welcome, everyone. I see folks starting to trickle in. We're going to give it a minute. While we're at it, good morning, good evening, good afternoon. There's people joining from all over. All right, let's get started. So again, welcome everyone. This is so exciting. Welcome to today's TileDB Tech Talk. I'm very happy to bring you our March edition. My name is Devika Garg. I'm the Director of Product Marketing, and I'm joining you from California. Also joined by my colleague Maher Hamdi, who's ensuring smooth operations for this webinar for the next hour that you are with us. Today, we have the privilege of hearing about spatial transcriptomics from not one, but 3-4 experts in this area. So first off, we have Max Lombardo. He's a senior product application scientist at the Chan Zuckerberg Initiative. And we have Peng He, who is assistant professor of pathology at the University of California, San Francisco. And then we have our very own Aaron Bullen, who's a staff product manager for single cell solutions at TileDB. Hey, Aaron. And last but not the least, we have Julia Dark, software engineer on the single cell team at TileDB, also here to answer questions as we go. So thank you, everyone. We're so grateful that you chose to spend this next hour with us. It's very great to have all of you speakers here. Spatial biology is in general a very hot area, upcoming area of research, and with very complex data needs. So we're going to hear all about that today. Let's see. So before we go in, that's us. That's the title of our talk. So before we go in to the talk itself, we'd love to get a quick level set of, with 3 questions to get gauge an understanding of where you are with spatial data today. You may be a beginner, you may be deep into the weeds. This will help to our speakers. So if you could please take a few minutes. And any thoughts about these poll questions, Max? I know you shared a few of these. Any thought process around this?
Hey there. Yeah. So really, we just want to learn about how people are starting to use spatial in their workflows. I think, it's a technology that's been around for a few years, but I think in this year in particular, it's going to see a big uptake and already has seen a big uptake. So just really looking at how people are accessing this data currently, what are the challenges in the data landscape and the tools that kind of surround it. And yeah.
Nice. All right. So there we go. So we have some answers. So primary interest, as we can see, there's many areas where you could apply spatial. And I see a big chunk in multimodal data integrations, or spatial with single cell RNA-seq. A lot of beginners, actually, about 40% who are starting out. So they're not honed into any specific area. Then second question was more about challenges in data. Any thoughts there? So we're seeing these two on top, data size and integration with the existing pipelines. Probably something we'd expect. We're seeing. And then third question, how do you typically access and analyze spatial data? Custom scripts. Any surprises? Anything that's surprising at all? Or it's all kind of expected? Aaron?
No, I think this sort of reflects the diversity we've been hearing from a lot of our customers and partners, that It is a new area. A lot of people are sort of at the exploratory level. But I think, the one thing that is consistent is people are sort of universally interested in starting to lean in more to spatial than they have in the past.
Wonderful. All right. Thank you, everybody, for your responses. I think this is our cue to dive in. So I'm going to be moving the slides, and I can transfer control. Aaron, so would you like to take us through the agenda?
Yes. Okay. I just requested control.
Great.
Oh, there we go. Perfect. Yeah. So today's webinar, like Devika said, is all about spatial. We're going to talk about the new spatial capabilities in TileDB-SOMA, which have been developed in collaboration with our friends at CZI, and was leveraged to support the construction of the first CELLxGENE Discover census. I have to make sure I get that full name correct. So Max is going to talk about the new spatial census and how to access it. And we're super lucky to have Peng here, who's going to anchor the discussion and present some real world examples of spatial analyses that he's performed using data generated in his lab that's now available through the census. Okay, so why spatial? This probably isn't anything we need to sell this audience on. I'm sure spatial is an incredibly hot topic in the life sciences. It's probably why you're here. But this is something we've been hearing a lot from our customers and partners. And I think it's clear that spatial data is increasingly the go-to modality for studying complex biological systems. And of course, the big differentiator there is, unlike traditional single-cell approaches, spatial technologies maintain the spatial context of their measurements, which greatly expands the types of questions that can be investigated. And the benefits of spatial data are being realized across many different domains, from the role of spatially variable gene expression on tumor heterogeneity to mapping neuronal interactions in the brain to study behavior. There's just a lot of really exciting work happening in this space. And you'll get to hear a really interesting example of this in a moment from Peng, who's going to talk about his work studying limb development, which is where this beautiful image on the right comes from. The flip side of all this is that spatial data is much more complex than traditional single-cell data, at least from a data management perspective. And that is where TileDB-SOMA comes in. Oh, I think I botched that animation. That was cool. Sorry. So TileDB and SOMA, I like to start off, as I typically do in these tech talks, with a little bit of background, just to set the stage for the rest of the webinar and make sure we're all on the same page. So if you're not familiar with us, TileDB is a data management company that offers end-to-end solutions for life sciences research and discovery. We are actually on the verge of releasing the next major version of our commercial product, which we're calling Carrara, that provides a unified platform for managing and analyzing all of your data. It allows you to store and organize all of your files, regardless of format, with one unified data catalog. It includes a serverless compute framework for running all of your analysis pipelines at scale. There's hosted Jupyter notebooks that are easily shared, and a really flexible permissions model for creating collaborative workspaces. And much of this functionality is built on our core technology, which is the TileDB storage engine. And we've covered TileDB Core in more detail in previous webinars, which I would encourage you to check out. But I just wanted to quickly highlight some of the key features of TileDB Core that make it a great fit for both traditional single-cell and spatially resolved omics data. So first of all, TileDB Core is a multi-dimensional array engine that natively supports both sparse and dense data. And of course, for spatial data sets, they typically include a mixture of both, right? So we have one solution that works equally well for count matrices, which are highly sparse, as well as images, which are typically dense. Second, TileDB is designed for high performance and scalability. It uses columnar storage that allows for efficient compression and indexing, which is critical for large data sets. And all of it's implemented in C++, so it's super performant, It can be used with a variety of programming languages, including Python, R, Go, Java, and many others. And TileDB is cloud native, which means you can store your data in the cloud and then access it from anywhere without any additional server infrastructure. So this means you can take advantage of highly scalable and cost-effective cloud storage from providers like Amazon with their S3 offering. And so that brings us to SOMA, which is our open source specification and API for managing single cell and multi-omic data sets. And SOMA is built on top of TileDB Core. There's a link here to the open source specification on GitHub if you're interested in learning more about it. And also, SOMA is a project that we designed and built in collaboration with CZI, with the ultimate goal of providing a language agnostic, highly scalable format for single cell data. Here's a look at some of SOMA's key features. So first of all, because it's built on TileDB Core, SOMA itself is language agnostic. We're not tied to any single programming language or framework. Currently, we provide APIs for both Python and R, which are the two most popular languages for single cell analysis. We also provide interoperability with a variety of popular single cell toolkits, including ANData and ScanPy, Seurat and Bioconductor. So you can store your data once and then access it from any of these tools. And of course, being built on TileDB means that SOMA also works natively with remote object stores. So you can easily query and filter your data directly from the cloud without having to download it first and load the entire data set into memory. So before we dive into the specifics, a lot of fun animations of our new spatial support, I wanted to highlight some of the challenges that come with supporting this data. So first of all, spatial experiments produce a variety of new data types, including images, point clouds, and polygons, which are not typically found in non-spatial experiments. And these data often come in different formats, which adds to the fun. Second, spatial transcriptomics is a rapidly evolving field, both in terms of the technology and the analysis software. So when it came time to design an extension to SOMA's data model to accommodate these new spatial data types, we tried to make as few assumptions as possible in order to maximize flexibility and extensibility. And so, with these challenges in mind, let's talk about the current scope of TileDB SOMA spatial support. Also keeping in mind that this is only our first release and we're actively working on expanding the functionality. So right now, our primary focus is on Python. Our support is on the roadmap, and if that's something you're interested in, please let us know. Feel free to reach out to me directly. In terms of the assay support, we initially focused on spot-based technologies like 10x Genomics, their Visium product, and SlideSeq. And the rationale for that was, these are widely used and there's a critical mass of these data sets already out there in the community. So that seemed like a logical starting point. And as far as ecosystem interoperability, we decided to focus on the spatial data Python package from the SCVerse team, which is where we have seen the most demand. And if there are other toolkits that are important to you, again, please let us know. Okay, so let's look at the data model. If you're familiar with SOMA, you know that the data model is based on a hierarchical structure of collections in arrays and includes designated locations for storing all of the data elements that are typically associated with a single cell experiment. The new spatial extension is entirely additive. So as you can see, we've added a new spatial collection to the root level of the experiment, which is highlighted in pink here. Inside of that spatial collection, we can store one or more SOMA scene objects, which is the primary organizational unit for spatial data in SOMA. And we'll take a closer look at that in a moment. And then on the left here, we have a table called OBS spatial presence. So that's used to map observations to specific scenes within your experiment. And this is only necessary if you have an experiment with multiple scenes, which might map to multiple tissue slides with an experiment. Okay, so SOMA-SCENE is our first new spatial class. It's basically a specialized SOMA collection that's designed to group together related spatial objects that all share a common coordinate space. Within a scene, we have the following predefined collections. Image is a collection of multi-scale image objects. So this is where the imaging data is stored. OBS-L is a collection of spatial data frames which contain the location information for the experiment's observations. So if you're working with a Visium dataset, this is where you'd store the spot locations and spatial metadata. var-L is the variable equivalent of OBS-L. So this is where you would store spatial data indexed by the features. And this isn't relevant for a Visium dataset, but would be for like an imaging-based assay, something like 10x Xenium. Okay, so let's zoom in on the multi-scale image class first. So multi-scale image is itself another collection that's comprised of SOMA dense ND arrays, each of which stores one resolution level of the images. So this is very much like a pyramidal image in other formats. For a Visium dataset, typically you would have the full-scale resolution image and then the high and low resolution downsampled images. And these would all be stored in one single multi-scale image. For the imaging metadata, we follow the OME and GFF spec as closely as possible to maximize compatibility. And all of that metadata is recorded directly on the collection. This is a feature of TileDB Core itself that we're leveraging. To access multi-scale image data, I've got a quick example here that shows how you would pull the low resolution image from our experiment into memory. So the first thing we need to do is open the experiment and access the scene within the spatial collection. Within the image subcollection, we can access the multi-scale image that contains both the high and low resolution 10x Visium images. And then we use the read spatial region method to actually load that data into memory. And again, in this case, we're specifying we wanted the low resolution image. Like all SOMA classes, the read method loads data into memory as an arrow object. In this case, it's a pi arrow tensor, but we can easily convert that to a NumPy array. And then here I'm visualizing the image using matplotlib. The next new class is the point cloud data frame, which is used to represent a collection of points in a multidimensional space. And so the main difference between a point cloud data frame and a standard SOMA data frame is the point cloud data frame must be indexed by spatial coordinates and contain some metadata about the coordinate space. So for a Visium dataset, we're indexing the tissue spot locations by their X and Y coordinates in pixels. And like any data frame, you can also add multiple columns to store additional information about those points. So in the example on the right, you can see we're following a similar pattern to what we used for the multi-scale image example. We first open the SOMA experiment and access the scene within the spatial collection. And now, because the cell spots are indexed by the study's observations, the data frame is stored in the OBS-L collection. So from there, we can access the point cloud data frame called LOC and use the same read spatial region method, which allows you to provide either a bounding box or an arbitrary shapely shape to subset that data. So in this case, we're handing over coordinates for a bounding box just to read in a subset of those points into memory, and then finally converting the resulting pi arrow table to a pandas data frame. Okay, so how do you actually convert your spatial data to a SOMA experiment? Currently, we provide a supported ingester for 10x Visium data that was generated by Space Ranger. So on the left, you can see a directory structure of the Space Ranger output files. If you've worked with 10x Visium data before, this should look familiar. We have our 10x HDF5 file containing the count matrix, and then all of the spatial data in the spatial directory. To perform the ingestion, we need to provide the URI where the new SOMA experiment will be created. In this case, we're handing it an S3 URI because we can write to remote object stores natively. The input path needs to point to the directory containing the Space Ranger output files. And then we need to give names to the SOMA measurement. For transcriptomics, this is typically RNA. And we also need to name the SOMA scene. From there, the ingester will take care of the rest and then produce a spatial SOMA experiment. And as I mentioned earlier, in terms of toolkit interoperability, we're focused on the spatial data package. And SOMA provides a convenient way to export a spatial data object from a SOMA experiment or a slice of a SOMA experiment. And so you can see the code here is pretty straightforward. We're calling the toSpatialData method from soma.io's spatial submodule. And we just need to provide an open SOMA experiment, the name of the measurement, and then the name of the scene. And the result here below is just a typical spatial data object. It contains all of the relevant data elements from the SOMA experiment. So you can continue on with your analysis using any of spatial data's APIs. And, again, this is a work in progress. So if you have any feedback about the APIs or, other toolkits that are important to you, again, feel free to reach out to me directly. We would love to kind of chat about this and learn more about the use cases out there. For now, I am going to stop here and hand the baton to Max, who's going to tell us more about the spatial census and provide some examples of how you can use it.
Thanks, Aaron. That was awesome. And yeah, let's see. I just requested driving permission here. Okay.
Let's see. I just stopped. Are you in control?
I don't think so yet. Let me see. Let me try requesting again. Oh no, it says I'm controlling. Okay, cool. Arrow keys work. Cool. So that was super informative, and I don't know. I really appreciated hearing Aaron spell that out, because I think it's the work that the TileDB team has done, and collaborating with CZI to build TileDB-SOMA has been really awesome, and it's been enabling, I think, a lot of really cool data access, right? And so, CELLxGENE, just to give an overview, at its core is this large collection of single cell and spatial data that is standardly curated. So more recently, we basically hit a major milestone within this corpus, and we crossed that barrier of 100 million cells, mostly composed of human and mouse. It's submitted by the community. So basically, we're sourcing from, yes, our grantee network, but really anybody who's generating high quality data sets and doing groundbreaking science. We're receiving submissions from any of those scientists. And here's a number of the top tissues, but I'll skip over that. Another piece that I mentioned is the standardization of this data corpus. So when we do accept data for submission, we require that it meets some structural constraints. So obviously we accept the data itself. So raw counts, typically normalized data. We standardize featured data, so linking feature names, gene symbols to ensemble IDs, for example. And then we also standardize the universally available cell metadata. So this can be things such as cell type, which we link to an appropriate cell type ontology, tissue, ethnicity, age, et cetera. For more information, you can go check out our schema. It's publicly available, so you can see what the requirements are and see, what the expected structure of any data set that's pulled from this census resource, what the expected structure will be. And so built on top of that large data resource, we actually have a number of applications. I won't talk about our visual tools today, but if you just want to go ahead and poke around and get a really quick glance at some of the data sets that are in the corpus, you can go to cellbygene.cziscience.com to be able to browse and download data sets, explore individual data sets. And this is another piece that I want to call out, where TileDB really helped us define the sort of back-end data piece to make exploring individual data sets performant. We also have the ability to sort of do meta-analysis over this corpus, so looking at gene expression across all the data sets that are there in a visual interface. And then we can also learn about different cell types in our cell type knowledge base called CELLGUIDE. But yeah, talking that, I did not program that animation in. Let me see.
I think pitch has like a magnifying glass
Yeah,
feature
yeah,
that I kept hitting.
Yeah, I think I'm discovering. Cool, thank you. So digging a little bit into the part of the Cell by Gene Discover suite that we are going to talk about. So on top of this data corpus, then we also have an API, right? And so basically this is Census. So Census is this data object built on TileDB and TileDB-SOMA, an API, which gives efficient access and also programmatic access to I don't know if we're the largest aggregation of standardized single cell data, but we have an openly available schema and it's pretty big. And I think, yeah, at any rate. So, but all of those things are in motion right now. At any rate, we provide programmatic access to this really like stellar single cell and spatial genomics resource. And the way we do that and how it sort of fits into the TileDB and TileDB-SOMA piece that Aaron was talking about in detail in the previous section of the talk is that we start with all of these individual submissions. So ANN data objects or spatial data objects that are submitted by authors individually, and then we essentially concatenate these into a larger object. As Aaron mentioned, there are different objects for spatial and just single cell data. But generally, this pattern holds true for both of those modalities. And then what we have is not only this large data object, but then also the associated metadata. And so what this enables us to do on the cell by gene census side is to wrap up the data engine and provide nice ways to access and query very fine-grained slices or well-defined, essentially construct well-defined queries of the data that's in census. You can download these. So Aaron was talking about the interoperability to various data formats, including plugging into the standard single cell and spatial genomics toolkits ecosystems. And then we've also made some progress in constructing features for more like model builders and machine learning researchers by enabling streaming of this data so you don't have to load everything in memory. And let me see. Sorry, I hit the next, let's see. Okay, a little bit of lag. And yeah, so cell by gene census is really built on this entire stack of technologies that Aaron had talked about. So on the sort of the user-facing level, there's the cell by gene census, which is built upon TileDB-SOMA and the SOMA specification. And so spatial is actually a very new thing for us. So we released, I think, our beta access of spatial via the Cell by Gene Census back at the end of January. And so just talking a little bit about the motivation, I think Aaron covered it really well, but I would just add that not only does spatial provide this additional information, but I think the two technologies really just complement each other very well. So we can see the expression of the same gene. So all of these images are actually from Pang's study. So I'm just pulling out some of these data sets from the census, which we'll go over how to do that in a couple slides. But essentially, we're looking at the expression of the same gene across the single cell and spatial data sets that Pang and his group had generated. And we can see, obviously, that, in college, we can see specific gene expression in a specific cell type, but we don't see like the spatial organization of that cell type. And on the flip side, we also have, this awesome spatial information, but actually the sequencing spots that are there are not as fine-grained, right? They're not single cell level. Although I'm sure that's going to change, in the coming years, but we can use these two data types together to really inform each other and I think fill in the gaps, which is why it was a really natural choice to expand to for the cell by gene census side of things. And so we got this. So we'll actually, I think we have a good introduction to this, just reminding us that we have, not only our census data where the single cell data lives, the spatial census objects, but then we also have this metadata, just general metadata info. So we can start exploring the census there. And then just getting into actually, seeing at a sort of a tactical level, how do you start, opening the census, digging around it, checking out the various metadata that exists there. So looking at that sort of census level metadata, I'm zooming in on a particular table that's there, so the dataset metadata field. And so within this dataset data frame, you can actually access metadata related to all the datasets that are in there. So in particular, I want to call out that we can find out what collection a particular dataset is coming from, in addition to various other information like DOI, and then also the dataset IDs. And in particular, we also have access to the original dataset IDs that have been submitted, and that'll come in handy a little bit later in the talk. And sorry, I meant to say original dataset objects. And the way that we can access that is actually to just specify the version of the census that we want to use. So we can specify the date of the last LTS, or long-term stable release. And those come about every 6 months, with more intermittent releases that are not designated as stable. And then the stable designation is just, we make a certain guarantee to host that, that version of the census is available and hosted for a certain number of years. I believe it's 5 years. And so we have this really handy information about like what type of data is actually in the census at the moment within that particular release. And then we can start to explore the individual objects or the data objects that are there. So by doing that, we've already specified and opened the census, so we can just actually pull out the single cell and spatial objects here. I just want to call out that for each of those different data objects, we do have separate measurements, or sorry, experiments or measurements. I always mix up the TileDB terminology a little bit, but individually for both mouse and humans in each of those objects. And just going a little bit down into the inner workings of the spatial object, so we can just see that we can access this here. So census spatial sequencing, Homo sapiens. So we're taking a look at the human data, and then we have just the various metadata associated with it. So we'll explore a little bit about what the structure of ops looks like, var, and then we'll also just demonstrate this data export to spatial data. Cool. So just taking a quick look at the spot level metadata. So this is within the human experiment. We can see that we have access to all of the standardly available and curated metadata columns. So I want to note here that in many cases, authors will have sets of columns that do not map to anything that's available in our schema, or that isn't defined by our schema in a standard way, so like linked to an ontology. Yet we want to be able to, and so essentially via the census, that's the only metadata that is accessible just via a regular query. A little bit later in the talk, I'll show how we actually get the original author metadata. But right here, what we get in the object is just the standardly available metadata. So we see all of these different ontology terms, which are standardized. Cell type, for instance, is coming from the CL ontology. We'll also have the ontology ID that's available to us. So this is quite handy in case we want to start to maybe group things into different lineages or different parts of the cell, quote unquote hierarchy. And essentially you can pick and choose which columns you want to actually pull down and view, but this is kind of everything. I think a couple of spatial specific columns. So one in particular is this in tissue column, where you can actually get annotations as to which of the sequencing spots in the slide are actually covered by a tissue, or actually measure a tissue, and which are just there and unused. Exploring the metadata a little bit further, we have our variable metadata. So I don't think anything super specific to spatial here, but you wanted to note that we have standardized feature metadata. So, we have a bunch of, feature metadata that's here, but I think, the interoperability and, emphasizing interoperability and making these features available with ensemble IDs is, definitely key for maintaining interoperability throughout the ecosystem. And then here we can see actually how we can start to construct queries. So, part of the reason I showed you all that metadata there is to motivate the fact that we can actually construct queries based on all those metadata values. So for instance, if we wanted to view essentially data that was associated with this particular tissue, we can go ahead and do that. And I've constructed this query such that, we're only going to return Peng's data right now. So this is good. Peng's currently the only contributor that's contributed hind limb spatial data here to the CELLxGENE census at the moment. So that was fortunate. But yeah, so we can specify which tissue we want. We could also combine filters. So we could specify tissue. And I think this case, like developmental stage would be quite relevant to combine there. And then we can also specify which column names we want to get. And I want to emphasize that this is just to sort of pull down the metadata table itself and start to filter and see how many entries there are. And I think this can get a sense of, how we can construct a query to get the right data that we want when we want to export the full object, because that takes slightly longer. So earlier I mentioned that, via the sort of standard metadata or OBS metadata field that's within each of those single cell and spatial census data objects, we only make the standard metadata available. And so I just wanted to call this out because it's kind of a common thing and relevant to Peng's study, but we do have a method of being able to retrieve the author metadata. And the value of that is that it's just much more flexible. It's a lot of that metadata is much more specific to the particular study and gives a lot of insights there. And I think in particular, being able to pull out, the author annotations in this particular example adds a lot more detail that I guess we'll hear about later. But I just want to call out that we can use download source H5AD to be able to pull out that original submitted object. And we can sort of just do that by identifying the dataset ID from the metadata tables that we've already examined here. Yeah, and finally, we have the ability to export to the spatial data ecosystem, which is part of the SCVerse ecosystem. And again, we're performing a query here. Actually, this time I'm performing a query with a slightly different access pattern. So here I'm using experiment access query. So we can pull out the specific experiment from the census, and then we can construct our query here. And again, this pattern should be familiar where we're specifying this tissue. And this time we're actually going to pull out the data itself in addition to the relevant metadata. And then just the other thing to mention is this method here, to spatial data, where we're actually performing the export. And then from there, you'll actually get a spatial data object that contains all of the different data sets that met the requirements for your query. So in this particular one, we've got 8 different Visium data sets that are all in the same object. So it's quite handy actually for bundling a large collection of Visium experiments, I found. And so, I'm going to pass it over to Peng to give you the real interesting stuff and like the really cool data that he's developed. But take it away, Peng.
Thanks, Max. Yeah, and so if you don't mind, so you can use your cursor, and I will just present over your slides. So to me, actually, all of the previous part of the presentation is equally exciting and interesting. And so for us, we are interested in the problem of limb development. For one reason, due to a lot of previous literature. That provides a lot of knowledge for us to better interpret the data. But also, the limb development has multiple stories that are related to the spatial patterning and the morphogenesis. So that is really an ideal platform, ideal paradigm to use a spatial platform and combine the different disciplinaries. And during the limb development in early stages, while it is still like above cells called the limb buds, there are already a lot of spatial domains or signaling centers that are being established. And so we know that a lot of these are controlling the anterior-posterior polarity and also the future axis establishment. So as the limb bud grows, so there is gradually establishment of the spatial axis, such as the proximal distal axis, And that determines the major identity of the three segments of the limbs in the mammals and a lot of other species. So if you look at the panel in the middle, in the center of this slide, in the past there are multiple models trying to explain how the proximal distal axes have been established, and there has been the progress zone model where the axis is sequentially established, or there is an early specification model. So this represents a second element of the spatial patterning. Well, for the distal part of the axis, there is also an added component of the spatial problems, which is the digit formation. And as you know, our digits on the hands and feet, they are not formed from the sprouting process, but rather a sculpturing process. In other words, for the cells that are on the foot plate, some of the cells will survive and keep growing, and they will end up in the future digits. However, for the other cells in between them, they will go through the apoptotic processes, and eventually they are doomed to death. And they will be giving away and to form the space between the digits. And so altogether, there are a lot of interesting questions we want to answer. And therefore, in the next slide, you can see. So Max, can you go to yes. So we really did a lot of efforts. So during my postdoc work in Sarah Teichmann's lab, actually in Cambridge, to map the cell types, cell states, and where they are in the whole process of human embryonic limb development. So this work has been a joint effort led by my postdoc supervisor, Sarah Teichmann, and the lab, supported by a lot of other labs, such as Omar's lab, Humboldt's new lab, and Barber's lab at Caltech, and a lot of other labs. And especially this, when we are looking at this data, and we also should appreciate the contributions from the donors. So without them, we will not be able to really capture a lot of these cool events that are happening during the human developmental processes. So how did we generate the data? So I want to spend a little bit time in the next slide to show you the design of the experiments. So as we discussed before in the previous slides, so this is still like spot-based spatial technology from the VCM Classic. And so what we did was to collect multiple time points during the time course of the early embryonic limb development, covering the early limb bud stage to the more mature stage, where you can see a lot of the lineages that are evident there. And below, in the lower part of this map, we are just showing some representative domains of these different limb samples. And so using this platform, and we can really quickly answer a lot of questions. And also because all of these data are already deposited, so for people whoever are interested in our data sets and to check certain genes or to validate certain hypotheses of yours in our data sets, you can easily download the data and make a lot of plots. So in the next slide, so basically, this is how our data will look on this platform. And when submitting the data sets, so we already integrated with a lot of the standards, thanks to the CZI, the CELLxGENE team, and also Lattice team, who helped to transform the data formats into a compatible uniform formats that can be easily curated using this system. So one example of using the system is that so you could inspect individual gene expression patterns using our data sets, for example. And in the next slide, we are just showing you one gene's expression pattern, which is collagen 2A1, and that is a broad marker for the cartilage lineages. And what you are looking at here are some early hind limb, including the foot plate sections. And so I think the yellow and red spots are the hot spots for this gene. And I think if you look at the sections in the in the fourth or the bottom right panel. So you can see, if you look clearly, you can see that actually there are 5 fingers highlighted by the high expression patterns of collagen 2A1, and that can give you a very initial expression of where the cartilage is forming in the embryonic limb. And of course, we can do something more sophisticated than this. So I guess you probably were wondering why Max was talking about like data and metadata, and also Aaron introduced the infrastructure. So why do we care the data and metadata? So actually, the funny thing is that the metadata can not only be like used to inspect a lot of QCs and a lot of other directly measured or logged metadata, and due to the structure of the data formats, the metadata can also be used to store a lot of the derived data or scorings. So for example, in the next slide. And so what I'm showing you is the scores or the percentage or abundances of individual cell types after the deconvolution process, combining our single cell atlas and the spatial data sets. So using a lot of algorithms such as the cell2 location here, we can derive the cell type specific abundance scores. So basically, we will know where each cell type is mostly distributed. Here, we are showing you just 6 cases. The first one, ADH fibroblasts. Initially, we didn't know what this fibroblast is about, but if you map them using the metadata slot and see where are the hotspots and comparing with another cell type, which is on the second panel in the first row, the MYL3 myocyte, and we can immediately recognize the co-localization patterns between the ADH fibroblast and the MYL3 myocyte. So this basically shows us that this type of fibroblast is mainly like overlapped with the muscle region. So this is probably a muscle-associated fibroblast cell type. And different from that pattern, we can see the articular chondrocyte, the third cell type, is mainly enriched around the knee area of the limb, which makes sense corresponding to the terminal part of the bone. Spatially. And we also have seen some weak signals of the fibroblast that is overlapped with the nerve, the sciatic nerve region that are able to be captured by the H&E image, and superimposing these two together, we know that this fibroblast is mainly associated with the nerves, so we call it neurofibroblast. And in addition to that, we also can see skin-specific cells such as the basal cells, and the hotspots are mainly on the edge of the tissue, which is the 5th panel here. And the 6th panel is also an interesting case, the hypertrophic chondrocyte, which is representing the terminal the state of the cartilage development. And due to the limitation of the spatial transcriptomics platforms, we are only able to see it in the broken part indicated in the center of the bone, and which is actually like consistent with what we know before. So, and overall, these examples are just showing you that using not only just data for gene expression and inspection, but using the metadata slot, we can look at a group of genes or deconvoluted cell type abundances. So, and I would envision that if you have a list of marker gene signatures, you can also calculate the scores of the list of genes and then store the scores in the metadata, and you can easily inspect the metadata, the scores using the metadata functions in a similar way. So I guess these are just some simple demonstrations using our data, and I will hand it back to Max to conclude.
Ooh. Am I sharing my screen there, or is that okay? Cool.
Yeah.
Okay, and yeah, for some reason my Zoom doc is gone, but that was awesome. That was very cool, and I think it's really great to see. I think the stack of technologies, and then census and like what it can, the science that can actually be applied for, and I think what level of science that it allows for access to. Aaron, I don't know if you wanted to say anything specifically about the TileDB, but I think
No, I really appreciate both you and Pang joining today. That was incredibly interesting. Someone asked if there would be links to the code shown today. And I did answer there. I just want to broadcast more widely that we will send out a follow-up email to everyone who attended that will include links to relevant documentation from TileDB that includes all of the same examples I showed today, as well as the super cool notebook from Max and some of the examples from Peng. So all of those resources will arrive in your email.
Absolutely. Thank you, everybody, for presenting. It was a very good talk. We do have a few questions in the Q&A panel, and if you have more questions, please feel free to drop them here. 3 questions actually around supporting additional spatial assay types. So asking specifically about support for 10x Xenium, Visium HD. So Aaron, do you want to take that question?
Yeah, sure. So love the question. Right now, we are really focused on spot-based assays, like we said, and there's still some work to be done there to round out that functionality. But we are absolutely interested in continuing to expand our support to include other spatial assays. In fact, we are already pretty far along on supporting geometries and polygons, which is a prerequisite for supporting imaging-based assays. Now, I will also say we are a very customer and user-driven company, so we're constantly trying to assess demand for different assay types and prioritize accordingly. So questions like this really help us do just that.
Absolutely. We have another one around ML, so data standardization. So could we use ML techniques to standardize data sets that are coming from, I guess, multiple small experiments and from different species? And how are you doing this already? Are you using ML techniques? Perhaps put another way, Max, if you'd like to take that.
Yeah, and I think Peng can probably speak to a little bit more to like cell to location, because I think that's a really interesting sort of like a data integration almost between the spatial and the single cell. But on the cell by gene side, so we're definitely piloting and exploring some of that, and we've made various investments into expanding our AI and ML capabilities actually as part of sort of a broader scope and broader mandate with our virtual cells sort of initiative. But on the cell by gene side, the progress that we've made towards that, I think, is, working with model builders to train various biological foundation models on the cell by gene data corpus. And, some of the outputs from that have been instrumental, I think, in some of the pilots of like experimenting with new, more automated ways of labeling. And so, just kind of like at a high level, right? Like having a foundation model which can find a good representation or embedding of all the cells, and then being able to pass a new data set that's perhaps being contributed or ingested, and like finding out how that maps to the larger corpus is something we're interested in. I think nothing in production yet, but I think in a fairly near to medium time frame, there will be some, how to say, additional automated capabilities to our data ingestion process.
Um,
Sorry, Peng, did you?
Yeah, I can add a bit. So I think for now, at least for my previous postdoc lab and also my current independent lab, it's so for standardization of a lot of data sets, it's quite challenging because I think for now we focus more on the interpretability, and there are always a lot of unexpected technical issues that we have to manually solve. But I think as we accumulate more experience, more ground truths, more training data sets, probably the ML can be more powerful. Also for now, I think the audience mentioned the species, which is a really great topic. And even for matching the species, it's also challenging, at least for the molecular level, and at what threshold we want to define the orthologs to a pair of genes to be ortholog. And there is really like a continuous spectrum. And also we want to make a lot of consensus decisions among the community. So we need a lot of back and forth and debates between a group of similar interests. And I guess the hope is that we can have a very smart ML to standardize everything. But I think before that, we are still like learning a lot of rudimentary stuff and also trying to settle the consensus between a lot of the biologists. So I hope that answered the questions.
Great. Thank you. Are PyTorch loaders available for spatial data objects?
So SOMA does have its own native PyTorch loader. Right now, we are mainly focused on kind of the classical transcriptomics use cases, and we haven't really started diving into the more specialized spatial areas, but it is absolutely an area that we are interested in pursuing. And even releasing the PyTorch loader in its current state is our kind of first step into that larger world.
Great. Here's a long one. So Jairaj Mathur, thank you for a great presentation. I'm curious about the normalization techniques used with spatial data. Is it a spatially aware normalization? And also curious about spatial data features in TileDB. Are these only available in the latest version? So I think, yes, the answer to the second one is yes, they are available in the latest version. And the first one, normalization techniques used with spatial data.
I can just comment on using our case. So I have to say for the normalization of the VCM datasets, it's very, very similar to the single cell RNA-seq. So what you do is that you take the raw counts after mapping the FASTQ reads to the reference, and then you get count matrices. And then you will normalize against total counts per spot, which is similar to the total counts per cell in the single cell RNA-seq. And then we do the log transform. So that's usually how it's done to accommodate the sequencing depths and also a lot of other basic metrics. And I know that there probably will be other ways of normalization, but as I remember when I submitted the data sets, it's already normalized on my end. So I think in theory, you probably could also normalize on your end, and also I think you could also choose to visualize the raw counts or normalize ones depending on the negotiation between you and the data submitter, I guess. But
Yeah,
maybe Max, you can.
yeah, I was gonna add, so for the data that's accessed in spatial, so when people do submit data, raw counts are required, and then normalized data is like strongly suggested. However, it's not a requirement. So you will find, some data sets just kind of like on the portal that perhaps don't have that layer. For access via the census, so for single cell, we actually offer both a raw count, expression matrix or data matrix. And then we also offer a depth normalized one. For spatial, I believe we don't have the normalized version. We just have that raw data. So, in making the image for this particular presentation, after I pulled the data objects out of census, I actually went and ran that, kind of regular or like single cell flavored normalization that Peng was just mentioning.
Wonderful. Let's see, I'm just gauging time and the questions. We have one more question on CELLxGENE, Max. Are there more examples of how CELLxGENE data is being used in the community?
Yeah, I think so. I mean, if anybody has like more specific use cases that they want to follow up on and see if it's possible to use CELLxGENE, definitely feel free to reach out to me. I think Devika TileDB will be sharing my email and contact information and the CELLxGENE team's information. But in general, I urge people to go to our website and check out. There's sort of in the top bar, there's a models page, and that showcases a lot of the work that we're doing with the community in terms of training large-scale foundation models and like really serving as a key resource in that particular area of effort in our field. And additionally, I've heard of so many examples of the CELLxGENE data corpus being stood up in, both public and private organizations. So, there's definitely that. I actually saw a lot of familiar people in the chat who I've chatted with before on that subject. So it's really cool to see the resource that's being used like just within the community. And one thing actually I want to dovetail that into, and something that I think we get asked a lot and we don't think to mention it, is that it's a totally open resource. So, you know, licensing is very permissive. And so regardless of the organization that you are at, you can feel free to utilize that resource and hit us up for help occasionally too.
Awesome. Thank you. I want to thank all the speakers. We have 4 more questions, which I want to get to, but I think what we'll do is instead reach out to you directly. I want to thank the speakers, Aaron, Max, Peng. It was really great having you here. And we do have this one last thing, a raffle. So for everybody who stayed until the end, we get to participate in a $100 gift card from Amazon. Max, do we have a winner?
Yeah, so for this month's raffle winner, it is a Jan Patrick Pet.
Awesome.
So we'll send you an email very shortly.
Congratulations, Jen. It's always fun. Couple of last items. So Carrara, Aaron mentioned our latest release, which is upcoming. You can please go ahead and scan this QR code if you like to join the wait list. And we also have our next month's Tech Talk coming up, which is going to be about population genomics data and running federated queries on it. We thank you for joining today, and we'll see you again next month. Thank you.